Hello, everyone, and welcome to TIDES Digital Week. This event is brought to you by the producers of the upcoming face-to-face TIDES conferences that are visiting Amsterdam, Kyoto, and Boston. My name is Lauren Berry, and I will be your host for today's session titled NextGen, a novel microfluidic solution for developing a wide range of nanoparticle formulations T. First, let me cover some quick housekeeping items. If you experience difficulties with audio or advancing slides, please refresh your screen by hitting F5. If you experience other technical issues, please hit the question mark button to receive assistance. At any time during the presentation, submit your questions into the Q&A window, which is on the left-hand side of your screen. All sessions this week are recorded. You'll be emailed in 24 hours when they are available for viewing. Now, let me introduce today's speaker from PrecisionNano Systems. Today, we are welcomed to have here today Tim Lever. He is the Director of Product Management. Again, thank you so much, Tim, for joining us today. With that being said, I would like to hand it over to you to begin your presentation.
Awesome, thank you very much. It's great to be here. Thank you for the introduction, and bring apologies on behalf of Dr. Ewan Ramsey, who wasn't able to join us, as you might have seen in the promotions there, but excited to talk a little bit about our newest platform and how this can be used to solve some of the challenges that we see people facing as they start to develop formulations for a wide range of drug delivery. So just to get us started and to put it into a frame of mind here, I'd just like to talk about why or what it is we're trying to do as a company. So PrecisionNano Systems, our mission is to accelerate the creation of transformative medicines that significantly impact human well-being. What I'll speak to today in terms of presenting some of the data and technology around this is really this idea of how can we accelerate the delivery of new medicines. As I'm sure anyone on this call can agree, it takes too long for too many patients to get even a hope of treatment. So really looking for ways to simplify processes and to simplify a lot of the manufacturing to make sure that that's not the bottleneck in bringing these solutions to people who need them. As we start to think about kind of the waves of modalities for treatment of diseases, we'd like to think about kind of these three generations. Small molecules are obviously the most predominant in terms of number. Dating back to the earliest days of medicine, this is what has been used to treat every disease that was treated, from very simple painkillers to more sophisticated small molecules. And it wasn't, as I'm sure we know, till the 1980s and beyond where the year of biologics has really come into full force. And we saw the emergence of technologies which enabled the large-scale manufacture of these biologics be one of the enabling technologies to these coming to market. And we believe here at PrecisionNano Systems that cell and gene therapy is going to be this third wave, which is going from chemical production for small molecules to cell-based manufacturing for biologics to these complex self-assembled molecules for gene therapies. We're already seeing this start to come to fruition in so many ways. So just looking briefly in terms of at least in the gene therapy space, we see last year the approval of Onpattro from Alnylam, previously called Tisaran, a small interfering RNA packaged inside a lipid nanoparticle to treat rare liver disease. And this has gone on to solve very well and to be a first treatment for patients afflicted with the indication. Likewise, a lot of excitement coming from Kymriah, the first CAR T cell therapy, and subsequent follow-ons, and Luxturna 2 from Spark Therapeutics, viral gene therapy for a rare form of blindness. And we believe that when we're talking about gene therapy and cell therapies, this is just the tip of a coming iceberg of a new wave of precision engineered therapies that are going to start to address diseases that currently have no way of being addressed. So first of all, we're going to talk about nanoparticles today and their use in delivery. So I wanted to take a step back and just, for those who may not be steeped in this field, talk a little bit about what they are and why they would be used. I'm focusing here on this slide specifically to a lipid nanoparticle. This is one carrying a nucleic acid, but a lot of the principles would be the same for a polymeric nanoparticle, which might carry various different payloads, or even a liposome, which would have an aqueous core, which could be used to load a small molecule drug. That would be a liposomal drug, such as Doxil or something else. But in this slide, we're looking at a lipid nanoparticle for nucleic acid. So what we see here is they're actually a complex assembly. We'll talk about that again in a second. But what we're seeing is there's a mixture of different lipids in there. And so we see a PEG lipid, which is the one coating the outside. These are often used for stabilization, both in terms of stabilizing them from preventing them from adhering to each other, stabilizing them through the processing in manufacturing, as well as providing some shielding in the body. So the general consensus is that this PEG layer is shed circulation, and so that increases circulation time that happens before the payload is delivered into the cell. Importantly, we see the green inside there being the nucleic acid. That's usually complex with an ionizable lipid, so this is a charged lipid, which is able to change its charge based on the pH. So during formulation, it's able to obtain a charge which allows the complexation to the nucleic acid to do the packaging. And then once it's in an in vivo setting, we can actually have a neutral pH so that we're not causing an immune effect from it. Inside there, we also see various other components represented there in the purple and the yellow. These would be structural lipids and cholesterol, which help give the particle its structure and its rigidity and its shape. Importantly, though, when we're designing a system like this, as I very briefly described there, all these components have their job, have their function within the particle, but also we're looking for them to have low toxicity, and that's been a big effort in developing these over the years, making sure that we're not causing an undesired immune response when these are being administered. And as I alluded to in the last slide, a lot of excitement has come from the approval of Onpattro, being as how that's the first clinically validated and commercially validated application of one of these lipid nanoparticles for delivery of a nucleic acid. And obviously the purpose of doing this is to protect in this case, the nucleic acid from any kind of degradation. If nucleic acid weren't protected, either through a lipid nanoparticle like this, or through a polymer, or even through some other technique, as we all well know, our bodies would quickly degrade that, and it would never get transported into the cell. We wouldn't get the therapeutic outcome. Mentioned there, getting uptaken into the cell. So not only are we protecting the nucleic acid and making sure that we're presenting low toxicity, we're also looking to have a system which will promote cellular uptake. These LNPs in particular can be uptaken by endogenous mechanisms such as the ApoE pathway, the LDL pathway that is taking up those molecules, and that's a big part of playing into the low toxicity, of course. And then lastly, in this complex design, one of the things we also have to design around is making sure that we get release of the payload from the endosome and into the cytoplasm. So talking about wanting to if we wanted to make one of these formulations with our payload of choice and our delivery system of choice, there's a good chance we'd be doing that for a rare disease. Might be doing it for an immunotherapy too, or even for a vaccine, which we can talk about. But let's just think for a moment about rare diseases. Right now, as many as 80% of rare diseases have at least a candidate genetic cause. A very high percentage of them have an identified genetic cause, up to 80% of them in some cases, and more than that are obviously candidating for further exploration. And what's exciting, of course, is that the gene therapies offer more avenues to treat these than conventional drugs, and offers an opportunity to treat the actual root cause. When we're thinking about small molecules, they can be very effective, but it's more through a serendipitous association. Two things interact in a way that prevent the eventual eventual phenotype of the disease. But with a gene therapy, the goal is to get in there to treat that root cause. In the case of RNAi, we can downregulate a gene that's coding for defective protein. That would be the case of Onpattro, which I've mentioned a few times. So in that case, we're delivering a small interfering RNA into the liver, which can turn off the production of protein, amyloid in that case. For an mRNA treatment, we can actually deliver messenger RNA, which would cause the expression of a protein or enzyme which might be missing in a particular disease. So we see this a lot of excitement for this in metabolic enzyme deficiencies. And likewise, in gene editing, we can permanently add or delete a defective gene. Deletion is generally considered easier for the state of the art, but I'm sure at the rate which everything is developing, soon we'll see additions coming as well, opening a whole new range of potential for treatments and possibly even curative administrations. But through all this excitement, there's great potential in the ability to treat diseases that currently have no treatment through gene therapies, and really getting down to a space where we can digitally design medicines and predict their efficacies up front is a significant challenge, though, in manufacturing. We hear about this a lot in the news lately with respect to viral therapies, where the predicted demand over the next few years far exceeds the global capacity to produce them. And it's not just a case of buying more bioreactors. Scaling manufacturing is a significant challenge. And even when we talk about non-viral systems, manufacturing raises a lot of questions. These are complex systems. And throughout, whether you're looking at reports from various different agencies, the constant thing you'll see cited is that manufacturing of cell and gene therapies requires significant expertise. It's costly. It's time-consuming. And a big part of that cost and that time-consuming is the ability to move between scales, the ability to say that something we made either in research, in development, or even in early clinical stages can then be scaled to a commercial stage. And we believe this is one of the significant hurdles the industry faces in bringing these treatments to patients is how do we get through manufacturing. Now, of course, one of the challenges when we're talking about a nanoparticle-based drug, one of the really challenging aspects in its manufacturing is that these nanomedicines are more complex than any previous generation of drugs. When we talk about aspirin, it's one molecule made up of about 21 atoms. A more complex small molecule therapy is still a single molecule. That might be 3,000 atoms. Looking at Herceptin, biologic there, it's estimated to be about 25,000 atoms placed in to make up that drug. But if we look at an RNA lipid nanoparticle, and these are estimates based on modeling, the estimates are that there's up to 100,000 molecules in there, and 17 to the 7 or more or less, an estimate there of the number of atoms that are inside one particle. And we're generating millions of these particles that each have to have the same characteristics. So we're looking for the same size, the same composition, the same charge of these very complex particles in order to get consistent results in manufacturing and ultimately a drug that can be approved by regulatory agencies. So the complexity that we're talking about here is really at a completely different level than anything that's been made before. The way these drugs are formed, if we have a bit of a screen grab from an animation that you'll see on screen. So this is a model that was done of how we believe these particles are actually formed. And so you'll see on screen is just a couple or 8 frames from a time-lapse series. So what's happening is you're taking these components that I described previously. In one case, we would take, this is again an RNA LNP that's shown on screen. So we would take that RNA, we would put it inside of a buffer. We'd take those other components, those lipids and cholesterol, and dissolve it in a solvent of some sort that's amenable to dissolving the material we want. So let's say ethanol. When we bring those two streams together, what's going to happen is that aqueous buffer mixing with that ethanol is going to cause all those lipids to fall out of solution. And as they fall out of solution, they're naturally going to want to self-assemble into their most thermodynamically stable form. So the charged ionizable lipid in that case will complex with the RNA. As those start to form, then the other lipids, based on hydrophobic hydrophilic interactions, will begin to form around that. And then finally, the pegylated lipid assembling into sort of core. And there's various different models that are being investigated as to precisely what the structure looks like. But really, the core message here is this idea of self-assembly. So all of these components want to come together. They want to form a lipid nanoparticle structure. And the goal then is to give them the opportunity to do that. And if we are able to make a process that's doing this mixing consistently, that's keeping the environment consistent, both from batch to batch, but also over time, so over time in one single run, then we believe we can get consistent particles coming out. The environment is changing, either over time, from batch to batch, or even in a local environment. So if we're making a batch, and for example, if we imagined we were doing this process by taking two buckets and pouring one bucket into the other and stirring, you can imagine there'd be all kinds of different microenvironments in there. And those different conditions would affect how this self-assembly happens. So what we want to do is we want to have a process that gets out of the way. We don't want our conditions to be what's dictating particle size. We want everything to be consistent enough and reproducible enough such that they're really becoming the formulation they want to be. So with all that said, one of the challenges when we talk about manufacturing is that the needs of different scale can be quite different. And so you can solve possibly for the needs of preclinical development, and you can solve for the needs of clinical manufacturing, but then how do you bridge those if those are two different solutions? And so conceptually, it's not hard to imagine how these needs are different. If we're thinking of doing these formulations for preclinical setting, the batch sizes are very low. We're going to be manufacturing them quite frequently. So in a day, we might want to do multiple runs to change different parameters, to change different components, to do different experiments or different screens. The regulatory burden would be non-existent to very low, possibly, as we get into a GLP setting. But we'd still want high consistency. So we would still want very consistent results so that we know that our experiments one day to the next between scientists and collaborators are consistent. Then if we think about a clinical setting, our batch sizes are likely to become quite high. Now, what exactly high is can be relative, and that's something I'll mention when we're talking about a population-based therapy for a rare disease. That could be quite high, quite in volume. But when we talk about a personalized medicine, which we could talk about immuno-oncology, could certainly be the case. You might be talking about one batch is one dose for one patient. But even still, compared to the research scale, it's still going to be a transition to a larger batch. The frequency of manufacture can be quite low in that case. You're probably not making multiple batches in one day outside of personalized medicine. If we're in a population-based indication, then we might be making one or two a year in some cases. But we do have a high regulatory burden. Obviously, all this has to be done under CGMP conditions and meeting all those stringent requirements. But again, our consistency is very high. So we want solutions that enable us to do research efficiently, enable us to meet the requirements of preclinical development, but also enable us to very easily transition to clinical development. The last thing you want to do is to have a promising therapy which doesn't make it to patients because of an inability to manufacture. That's really a tragedy. And so as this field has developed, the idea of drug delivery as a whole, starting with liposomes for small molecules and on, we've seen an evolution of technologies. So early on the left of the screen, we see high energy techniques. So these are the techniques that were first developed by researchers and scientists who were first experimenting with this idea of nanoparticle-based drug delivery. So for liposome delivery of small molecules, or polymer nanoparticles and other things like this, they were first understanding what these systems were actually capable of and their structures. They developed applications, or I should say techniques, such as sonication or extrusion, where you're basically generating some bulk population by either large-scale mixing or evaporation processes to create thin films, followed by rehydration. But the key thing that's happening there is you're taking some some initial product, some initial bulk mixture, and then doing a secondary high energy process to break it down. So in the case of extrusion, passing it through a membrane. In the case of sonication, applying energy to break them apart. Those are very harsh processes. They're inherently difficult to scale. The physics of passing a few milliliters through a membrane, you can imagine the local conditions there can be quite different, and the controls that have to be in place to do that well are very different than the process to do 10 or 100 or more liters in a batch. So there's a lot of difficulty with scaling these kinds of processes. And then when we start thinking about gene therapies, these are just simply not amenable to working with very fragile payloads. So as we saw the emergence of nanoparticles for gene therapy, we saw the development of inline macro mixing. So this would be basically where two streams are brought together and collided or mixed in some way, which creates a turbulent flow. So you see that represented in the image there. We get two streams colliding, get generally very fast mixing, but in a turbulent condition. So that's a chaotic process. It works, or it can work, for the development of RNA lipid nanoparticles. This is a kind of class of production that is used for Onpattro. But what we've seen is that this can be difficult to reproduce. So those chaotic conditions are difficult to control. So from batch to batch, we do see a tendency, especially at least in the development arena, for that to produce variations in results. And as we start to imagine getting to larger RNAs, so large mRNAs, really some questions as to whether or not the applicability is going to be there, or if this chaotic, high shear, uncontrolled process is going to damage the payload as well. We do see improved scalability, which is one of the selling features here, but we do see challenges with scaling this down. So the conditions, as we say, where the particles are actually being formed, that's what we want to preserve. That's what we want to try and maintain. But going from a very small T junction to a very large one, because it's a chaotic, uncontrolled process, getting that scaling back and forth be very time-consuming, can be very challenging. And so then what we saw was the emergence of microfluidic applications, and this is both a lot of work coming out of Harvard and MIT in what we would call flow focusing, which is the microfluidic approach there, the lower image, or in the case of the work that we've done here at PrecisionNano Systems, our early microfluidics, which are represented on top there. These approaches were originally developed with an eye to how can we make development easier for people in the lab. So how can we make something that's reproducible? So from day to day, person to person, even run to run, within one day you will get the same results. How can we remove the art from the process and really just make this something that's repeatable? How can we do it in a way that's amenable to small volumes? So in the case of macro mixing, you need relatively high velocities to make this work, and so inherently you can quite a bit of material. So with microfluidics, we're able to scale that down to make milliliters, or in some cases microliters material, and start to democratize this so that people who don't necessarily have experience in process development, don't necessarily have experience as formulation scientists, can begin to access the power of these technologies. But what we saw is that some scaling challenges remain. So in the case of flow focusing, those are very limited in terms of being able to get to milliliters per minute type flow rates. In the case of our classic microfluidics, as we've called them, using like a herringbone mixer, those are able to get to tens of milliliters per minute, which is a great lab scale. And there has been work to show that that can be parallelized, to then take that reactor, put it in parallel, and achieve therefore more throughput. That adds some complexity to the system. So we've been really excited to introduce NextGen, and I'll show you some data around this next few slides, where we've developed a proprietary microfluidic structure which is possible to scale. So we can take one single microfluidic reactor. We can do mixing at bench scale, so I'm talking 10 to 20 milliliters per minute, and get excellent particles coming out. But we can then rationally scale the dimensions and the configuration of that mixer, of that next-gen mixer. So we can now go to hundreds of milliliters per minute through one single mixer and get the same results coming out. So it's very easy to scale. The manufacturing of the microfluidics is simplified, allowing us to access a range of materials and also have a very easy path to compliance in a quality sense. It makes it possible to integrate multiple mixers in series. So as we start to discuss and imagine more complex formulations that might include things like targeting molecules on the outside, that's now possible. Everything is very reproducible, but most importantly is we're preserving non-turbulent flow inside of these. So everything that's happening in here is not a chaotic process, it's not turbulent, it's time invariant. So across time in one run and from day to day, we're going to get the same results. And so what we see here is a study we did to show that we can generate lipid nanoparticles carrying a messenger RNA. And we can do it in a classic structure. So this is shown on the previous slide. This is what we call a staggered herringbone mixer. It would have been found on the nanoassembler benchtop historically. It's something that's been published on quite extensively by many people in the field. We can produce an mRNA LNP carrying a luciferase RNA, and you'll see that in the gray bars there. We can also take that same formulation with that same RNA without changing anything about the formulation, produce it at roughly 20 times the throughput, so from 12 mils per minute to 200 mils per minute. And you'll see that the activity of these particles is preserved. So as we do the dose response and we see the images on the right of the animals themselves, we're not able to distinguish between the two mixers because we've made the same particles just at much higher throughput now. And it's not just about scaling flow rate, obviously. If you're looking to make a batch of something, you have to be able to make that over a period of time. What you see here, this is a liposome that we've manufactured, so an aqueous core particle. On the left in the gray, again, you'll see a reference sample that was prepared just on a benchtop R&D system using the classic microfluidics. And then in the middle bars in purple, this is taking it onto a next-gen mixer. So this is running at about 48 liters per hour flow rate with inline dilution, so 12 liters per minute, so 12 liters per hour pre-dilution. But you'll see from the 5-minute mark, 10-minute mark, 15-minute mark, all the way through a 30-minute run, the particles are completely consistent. And when we look at the bulk population, again, size, light dispersity, so basically the width of the size population is very tightly controlled. We can also take that through to downstream processing. So tangential flow filtration, TFF, is an important step in this manufacturing to get rid of any residual solvents and to do a buffer exchange, as well as an ultimately stellar filtration. We're able to preserve those particle characteristics. I'm going to go fairly quickly here. I won't go into a lot of details, but I'm happy to answer any question. We've shown originally there mRNA lipid nanoparticles. The same was true in the last slide you saw for liposomes. Same effect. It's possible to use this mixer to make plasmid DNA formulations at a variety of flow rates. The trends that we expect to see, so when manufacturing liposomes, if we look in the literature, the ratio of the two components, so that aqueous stream to that organic stream, changing that ratio can affect the size. We see that trend preserved as we go between the two different mixing structures, the classic, which is what's extensively been reported in literature to date, and the next gen, which we've now released. So very exciting that we're able to produce the same particles, control the outcome in the same way. Now we're able to scale that. Likewise, the same is true for a PLGA nanoparticle that the polymer may include a nucleic acid, or it may include a small molecule. In this case, they were just empty. But again, the trends in size control that were previously reported using the classic microfluidics are still preserved in the next gen, and we now are able to take that to larger scales. And so really, the reason we're excited to share this technology is because we believe it's going to be enabling to getting a lot of these technologies that a lot of investment has been made in researching and developing to date. We're going to give them a very straightforward path to clinical production. We'll talk a little bit more about that. But with all this in mind, I do want to take an opportunity to speak to one of the ways which we're doing that. I've spoken here about how we can go to higher flow rates and how we can go to higher throughputs. But at the same time, we're bringing this next-gen technology into the lab as well. Yesterday we announced our newest instrument called the NanoAssembler Ignite. So this is an instrument that's designed with the end in mind. It's designed for the future. It's really designed to maximize time. And a big reason for all of this is that it's powered by this next-gen technology. And I just want to speak a little bit to how we answer these questions about why it's designed and how this is going to help really bring these therapies forward. So when we say design with the end in mind, what we're saying is that we want to provide the capabilities for people from the very start of their development work to develop robust and scalable process for their drug. And the way we think about this, and the way we would encourage anyone to think about starting a drug development program, is really about unit processes. So we've done a lot of work in developing a GMP system that I described there that can run hundreds of milliliters per minute or tens or more hundreds of liters per hour. And what we've now done with the Ignite is we've said, okay, well, what are the components of that manufacturing process? How can we bring those down to the bench? So one of the most important processes is particle formation, where the two streams come together and particles are formed. And in the GMP settings I described, we're doing that with the next-gen technology. So we were able to take that mixer, scale it down, and put it in a cartridge that can work at bench scale. Then, okay, well, after we've made particles, what happens to them? Well, usually we found in manufacturing of these formulations, a dilution step is required. So particles are coming out of the formation process. There's still some solvents present, which might make them quasi-stable. So a dilution is required to stabilize those. But oftentimes, that step hasn't been considered until we get to large-scale manufacturing, but it can be an important step. How that's done can affect the particles themselves. If it's done improperly, it could destabilize them. If it's done properly, it might stabilize them in a beneficial way. So now with the Ignite, inline dilution is available on the small scale. And so you can begin to imagine how you can start to model final large-scale production process on a bench from the very start. This could then be paired with a TFF system. So from various companies now, there's TFF systems available that can do 2 milliliters or more, or very small amounts. And obviously, you can do sterile filtration as well. And so the way we're conceiving this and the way we're encouraging people to think is, on your bench right now, you could recreate all of these steps so that something that you do today, if it passes all of these, you can have confidence that it's going to be have an easier path forward. If you work on a formulation that can't get through these steps on the bench, it's probably not something that is worth pursuing at this time, or it needs more optimization so that it can make it through these steps on your bench, because these are the same steps you're going to be using when you start to get to scale. It's really a time now where we can say you don't need to substitute different processes. So dialysis was very common and can still be used for very low-cost downstream processing. But if we substitute that out, as I said, for a small-scale TFF system, we can have that greater confidence that the formulation we've worked on and the candidate drug that we want to advance is not going to run into a roadblock further down the line. Just quickly, I want to touch on some of the things we're excited about the future when it comes to this platform. As I said, one of the advantages of NextGen as well, other than the scalability that we've talked about here, is our ability to do and look at more complex systems because we have a lot more flexibility with these microfluidics. So we want to enable the creation of medicines that are beyond the generation of nanoparticles that are being discussed today. So this might, targeting a big application we get asked about, or coding. With the NextGen, we have that flexibility to really expand the offering of different innovative microfluidics that will allow the scientific community to be more creative and to start to play with materials and processes that aren't possible right now. And lastly, we're excited that this is going to bring unprecedented speed, both on a day-to-day aspect, as well as in terms of a complete process. So when we're talking about day-to-day, the Ignite itself is completely optimized to be faster than anything we've made before. But really, when we're talking about maximizing time, we want to maximize or minimize, I should say, in this case, the time it takes to get a drug to the clinic and to patients. So manufacturing should not be long and complex. We believe there should be predictability, that something you've made in your bench can be brought forward. And our goal is to give people the tools to make sure that they can have confidence moving forward, get their products into the clinic, and bring these treatments to reality. A big part of that is making sure that you don't need to be an expert from day one, so we can give you the tools so that you can develop and validate a manufacturing process that somebody else can carry on and finish development on. You don't have to be an experienced specialist to do that. So with all this said, just wanted to give a brief introduction today on why we're excited about NextGen, why we believe this is a technology that will allow you to go from microliter screening to full commercial production. So what you see on the screen right now is just an example of some of the instruments we've incorporated NextGen in. So it's shown all the way, or it's used all the way from our Spark, which can formulate microliters at a time, into the Ignite now for bench scale formulations, Blaze, which enables a lot of CMC and process development at the 10 milliliter up to 10 liter scale. So that incorporates now the NextGen microfluidics as well, all the way onto our scale-up system, which can be used for CGMP manufacture, and it's a modular system, so it can be configured to achieve all kinds of different throughputs, whether it's a personalized application or whether it's a large batch application. So with all that, I'd just like to thank you for listening, and happy to hand back over here to Lauren and take any questions that anyone might have.
That's great. Thank you so much, Tim, for an excellent presentation. We have received a few questions already, but I do want to give the rest of you a moment to think about and enter your questions into that Q&A box, which is on the left-hand side of the slide. So once again, before we begin the Q&A, I'm going to run through some brief announcements. First, I would like to thank PrecisionNano System for sponsoring the event this week. I would also like to draw your attention to the upcoming face-to-face TIDES conferences, where you can hear from hundreds of expert speakers and network with colleagues, just like Tim. TIDES this year, we have one more face-to-face event in Amsterdam, which is in November. And then in 2020, we will be visiting Kyoto in February, and then we will be back in Boston in May. Also, be sure to check out the downloadable resources in the resources box to the right of the slides. Now, let's go back to Tim, and we can begin the Q&A. So, Tim, we have the first question here. Can you give me an example of how the mixing design allows formulations to be made with targeting or other complex structures that can be made?
Yeah, that's a great question, and I'd say a few things around that. One is that targeting is is an emerging field, and there's different ideas of how that's going to be achieved, whether it's conjugation to the outside of a particle or whether it's post-insertion of, for example, like a PEG lipid that has a moiety conjugated to it or a ligand conjugated to it. So regardless of those, I think first of all, what has to be considered is that all these ideas remain true, that we need reproducibility, we need scalability, we need predictability, whether we're doing that conjugation, whether we're doing that post-insertion, whether we're doing a coating or anything like that. So we need to make stable particles in step one, and then we do the second step also in a scalable, stable format. So the reason we believe NextGen is very amenable to this is the ability of different flow rates, various different throughputs that we're able to achieve predictably, scalably, allows us to put, for example, mixers in series. So you could take, conceptually, you could take a very one particular configuration that works well for lower flow rates to do an initial particle formation step, and then depending on your chemistry, you might need to introduce a much higher ratio of a second stream to get high efficiency of your conjugation taking place. And so with the next gen, we have the flexibility to really pair things together, bring about controlled processes across a variety of different situations like that. So I hope that answers the question a little bit. It's really about the flexibility to get to all kinds of different combinations depending on the needs of the.
That's great. Thanks, Tim. There is a follow-up actually to that question. How many batches can be made per each microfluidic cartridge in terms of material, in parentheses, volume or mass, passed through the system?
It's a question that depends a lot on what the particular materials we're discussing are. So I'm going to answer this particularly when we're talking about commercial manufacture, because I think that's where where probably the intent is coming from, thinking about how big of a batch could be made. In that case, in terms of number of batches, oftentimes we want to keep that to a single batch because thinking about gene therapies, the cost of validating and cleaning, for example, is far too high, and so we designed the workflows to be compatible with single use across anything that's fluid contacting. But in terms of volume or mass, really we haven't seen a limit to how high these can go. So traditionally with microfluidics, and it actually gives me an opportunity to discuss something that I didn't have a slide on here, one of the concerns has always been around fouling. So you've got small channels, so there's always a concern about aggregation in those channels, and for a lot of lipid systems, it can be very present even at the small scale where channels become blocked or occluded and causing large pressures in the system and ultimately possibly the need to shut down. As we move to the next gen paradigm, we can generate bigger channels which have optimized fluid velocities such that we really minimize the amount of or let's just say the opportunity for anything to aggregate onto the walls of the system and really minimize the effects of that in terms of pressure rises. It's a batch. So in terms of volume, there's no one answer. It's something that can be optimized for every case. In terms of a lipid system, I kind of gave a very brief example early on of a plasmid formulation that we made. Plasmid, along with a very charged lipid, is kind of one of the worst case scenarios for fouling. And we've been able to generate batches in the liter range through a single microfluidic cartridge of those challenging formulations. And so we believe that we can configure this really to get to whatever size batch is required.
That's perfect. Thank you. Next question. What are you seeing nanoparticle drug delivery growing?
Yeah, it's really across a wide range of applications. Interestingly, we're seeing a lot of excitement, and coincidentally, this just came out a couple of days ago with some of the clinical data being discussed by Moderna. A growing number of people interested in vaccines for this kind of delivery. So packaging an mRNA inside of an LNP for infectious disease, and two for immuno-oncology. So the use of nanomedicines with RNA payload for vaccines is definitely a growing field, but really across the board. So as scalable processes have now become more known, we see a growing interest even in small molecule delivery. So whereas people would have shied away from liposomes before due to concerns about how that can actually be scaled and how reliable that is, we're seeing a resurgence of interest in those as well. So really across the board, from gene therapies to precision small molecule delivery.
That's great. Perfect. Let me see. We have another one here. What advice would you give a researcher getting started on a project which requires some sort of drug delivery?
I think the exciting thing about being in that position now is that there's a lot of tools out there to get you started, even if you're not an expert. And so I think the first piece of advice we would give somebody is just start. Whether it's one of the tools we've developed or whether it's various other resources that are out there. We're in an era now where we've gone from people needing to be specialists to people being able to access this technology. We talked about work that's been done to really democratize nanoparticle drug delivery. So we have discussions all the time with people like bioinformaticians who have designed some new payload and normally would have to build a team to even just begin to test it, who can now do very early testing themselves and then pass it on to a more experienced team to actually start doing the development, or a more specialized team, I should say, start doing the development. So first thing is just to get started. But the second piece is really consider process from the very start. So I made that reference to setting your work up in terms of those unit operations where you can test things from very early on to see how they're going to react to the different stages and steps of manufacture. It's a really powerful way to de-risk your project and to build both your own confidence, but also get external buy-in that you have a path forward.
That's perfect. Great, thank you. And we have another question in here. What challenges do people still face while, when scaling up a formulation?
So we're focused here in this discussion about how do we actually scale really that particle formation step. So how do it is a very crucial step when two things come together and you get that self-assembly. Controlling that well is crucial, and doing it in a way, referencing the previous question, that is not going to aggregate, not going to foul. So we've spent a lot of time thinking about that, but the truth is that's one piece, and so we're always talking to people and encouraging them to remember every step along the way. Again, referencing thinking about things in unit steps can be very helpful, but even with that breaks down even further than the 4 steps I showed. So how are you preparing your materials? How is that going to change from making a few milliliters worth to making even tens of milliliters or hundreds of milliliters worth? That preparation, how is that going to scale? How are you going to test that? Thinking about that at every stage. How are you handling your materials, and is that a scalable way? Is probably one of the challenges that we still encounter, and we still spend a lot of time talking about ourselves, is how can we make sure that every possible step is scalable.
That's great. Thank you so much, Tim. It looks like we have run out of time here. Tim, an excellent presentation. We really appreciate you coming to speak with us today.
Thank you very much. It was a pleasure.
If anyone did submit questions that weren't addressed, do keep in mind that Tim will reach out to you directly. This session was recorded, so you will receive a notification in the next 24 hours or so when the on-demand version of this session is available for viewing. Also, don't forget about all of those downloadable resources that are in the resources box to the right of the slides. And before you log off, please take a moment to complete the feedback form that will pop up once the session ends so that we can continue to improve your digital experience. On behalf of Connect 365 Life Sciences, have a great day.